trying to think of the one that I read recently about they traced the HIV back to the Congo.
Mm-hmm.
That was a very fascinating read. I think it's 6:10. I think we're ready to go. We're ready? Fantastic. So, Olu and Kevin, I want to welcome you again. Thank you for being here. We're in the wrong class. Boy, this is just like the first day of school. Oh, wrong time. You're coming back for the lawyer? Oh, okay. Well, you guys have them for 50 minutes. Feel free to ask any questions. But Olu and Kevin, I would like to ask you guys maybe to start off just by letting our audience here know a little bit about your background and who you are, what you do, and then we'll take it from there.
Sure thing. Thanks, Tim. I'll kick it off. Hey guys, I'm Kevin. So I'm a senior PhD student, which effectively means I've been in my field for several years now. I started off in high school biology, feeling like it was a really cool subject. It was my favorite class at the time. Wasn't really sure if it was what I wanted to do for the rest of my life. So went over to college, did a bachelor's degree in biology with a focus on biochemistry, just because I did really enjoy the chemistry side of things as well.
Where did you do your undergrad at, Kevin?
That's a very important question that I should have included. I went to a UC. So I went to UC Santa Barbara. And in addition to being where I met my wife, it's also where I fell in love with science on a brand new level. I was able to do research in a way that I think Tim has mentioned some of the current students at Mount Whitney can do with the academies. And so having that firsthand experience, being in a lab and seeing what that was, what discoveries were fueled by, the kind of day-to-day work that entailed, it made me decide, hey, I want to go for this, take it to an even higher level and get a PhD where I can manage my own project and create, hopefully, knock on wood, create my own discoveries. So that led me to where I am today. I'm currently working on getting my PhD at Harvard University in Boston. And the program is kind of a broad umbrella. It's a biological and biomedical sciences. And, it doesn't really mean anything in particular. But what I like about that is it gives a cancer biologist like myself a chance to meet and become best friends with really brilliant people in infectious diseases, for example, like Bolu. So I think that might be the best segue I can offer. Bolu.
What a great segue it was. I think one of the ways I like to think about Kevin and I's dynamic is that the things that he studies are when the body starts to attack itself and does things that it really shouldn't be doing in a normal sense. And the things that I study are when other things in the environment start to attack the body and really start to wreak havoc. I'm Bolu. I did not go to UC or grow up in California. I grew up in Nigeria, but I started and had a very different trajectory to Kevin. I approached medical research from a more people and patient-centered focus, where I started really with my bachelor's in anthropology at Swarthmore College, thinking about how people go through hospital systems, how they experience biomedical care, and importantly to me, how they experience infectious diseases, these things that across the global scale have a huge impact on economics or the ability to go to work or just like live life, farm, do things that are critical to daily life. But I also really enjoyed the experience of getting time spent in a lab, like Kevin said, really seeing what takes an idea to practice to a therapeutic. And so we both met at Harvard when I did my PhD. So I did graduate. I do have a doctorate right now, and I sometimes forget that I have that. But I'm also happy to talk about the infectious disease element of things and really doing that research and the current trajectory that I'm on now, which is how do you get that research to the people who need it the most? So I work a lot now in business development and really talking to biotech firms and people engaged in infectious diseases on how to get things from a lab, how to get them from a research institute into the hospitals.
Excellent. So how many years before both of you are going to, well, Palu, you have your PhD. Kevin, you're working on your PhD. How many years have you spent in higher education?
That's a good question. Paul?
My PhD was halted a little bit by COVID. And I have, if anyone wants to ask questions about COVID or what that looked like from a research perspective and not just the way we all experience that, happy to go into that. That was 5 and a bit years post my undergrad degree.
And Kevin?
And for myself, after undergrad degree, it's currently been about, as Paul mentioned, 5.5 years, just starting 6th year. And I'll hopefully be graduating in 2024. So that'll be 7th year. And that's mainly just because I work with a model that takes a lot longer in terms of like developing experiments. Mouse models of cancer tend to take on the order of anywhere between 4 to 6 months to do a single experiment. So, you know, they let us ask questions about humans, but they take a bit of a while.
And I'll mention to you guys, if you ever have any question, just let me know or say it and I'll repeat it so that they can hear it if there's anything you want to know in particular.
Yeah, please. I should have mentioned this actually in my bio. Like, you know, if at any point you guys have questions about something we mentioned, this can completely be a conversation. You know, we're happy to answer anything that comes up.
Yes. Right. She's asking in general, she's interested in becoming a pharmacist after high school. She wants to know what would be the path for that to best become a pharmacist. What was your name? Celine? Celine. Yes. That was Celine's question.
Thank you for the question. So I think we can speak to this a little bit because even though Bolo and I didn't study pharmaceuticals during our PhD, we actually were very close to an institution of higher education that did train pharmacists. And we even, Bolo, I don't know about you, but I actually did get a chance to know a couple. I don't know if you ever met any MCPHS people.
I did. And one of the things that I've come to really appreciate about modern healthcare systems is how team-based they are and how much focus goes onto the pharmacists who are providing that point of care beyond, I mean, there's the nurses that are plugged in, there's the PAs that are plugged in, but the pharmacists really have that knowledge to be able to dispense and make critical judgments in the way that people who aren't close to the patients can make. So Kevin will continue talking about ways to become pharmacists, but I have a deep and profound appreciation for them as they form the basis of what we consider modern healthcare.
Yeah, couldn't say it better myself. In terms of specific degrees, typically when you're in college, so typically you start, you know, go through high school, take your bio classes, have a good time, huzzah. Go to college and you can take a variety of different majors. Honestly, it's dependent on the college you go to. I think most of them, especially like mine, for example, had a specific major within the bio umbrella for a pharmacology degree. And I actually took a couple pharmacology classes, even though that wasn't my major, just because it was cool and interesting. So you would seek out those kind of majors, those kind of courses in college, and then apply to graduate programs. Typically, I think the very frequent one that you see on a pharmacist's resume these days is what we call a PharmD, which is essentially a doctorate in pharmaceuticals. And I don't know a ton of the details in terms of like how long those degrees take on average, but you could definitely look that up, you know, for schools in the area that give PharmDs and get those details. But those degrees typically involve labs and classes that you can take as part of your degree where you're, you know, presumably working with different drugs.
And I work with a fair number of PharmDs now, and what's really curious or very interesting to me about their training is how it can be blended into the undergrad experience. So there's a bunch of people I know who did either 4+1 programs, where it's a 4-year undergrad degree and then a 1-year appended to it that's tailored towards getting the PharmD, or they finish undergrad and realize that that's something they want to commit their time to, are able to do a 4+2 or 4+3. So as Kevin said, there's a bunch of options out there, but really at the end of the day, I think if you're interested about pharmacy and you're interested about getting to know what are the things that are being prescribed, what are the biological agents that are making a change in communities, that's an absolutely fantastic way to go. It gives you the patient exposure, but it also gives you that deep clinical knowledge of, oh, this is how this drug is acting. This is how this medicine is working in a way that you can't really get from other tracks.
Lena, what grade would you be in? So you're a senior. I was going to say, I know our VCIS has a Pharm Tech program where you can go to get your, basically you can work at a pharmacy as a Pharm Tech when you graduate, get certificated. I'm not sure all the details because I'm not a teacher at VCIS, but that is an option there. I believe they also have something at the adult school that you get your foot there if that's something you're interested in pursuing before you move forward to a 4-year. And then as far as the science classes, it's pretty much the same undergrad classes for any med major, I would think. Biology, chemistry, organic chemistry, and so on, microbiology, the core classes, if you will.
Yeah, I think the first like 2 years for any person interested in pharmacology are not going to look different from the rest of the bio majors. Again, like Tim said, all the same essentials: bio, chem, a little bit of physics. They force you to take it. It's okay.
A tiny bit of math too, just enough to be able to make those back of the envelope calculations. And I will say one of the things that I loved about high school biology and high school chemistry is some of the topics didn't seem relevant, or they didn't seem like they applied to what I was doing on the day-to-day. But looking back on it now, those formed the foundation for me to be able to do the kinds of science that I did or what I do now, like an understanding or a general understanding of what that pathway is, or what part of the body that's acting in, or like where this is important. Kevin and I have very different research interests, but the number of things that overlap in the day-to-day is surprising, given just how different they are, but also we had very similar high school trainings.
I couldn't agree more. That was a really fun question. Thank you for asking that. Anything else we can answer?
Do you have any questions? Because I've got quite a few. Okay, so Balu, what are some of the infectious diseases you're working with right now? I have to ask because obviously there's a lot in the news. So
yeah, so COVID is a big one. We're currently thinking about, and the way that I'm doing science now, we're thinking about how to predict the next new variants or the new waves, and what are the sorts of vaccines we should be making to prevent widespread calamity in the next few months, especially as we go into winter on the East Coast, where people are spending more time indoors and that all becomes a thing. But before that, I worked exclusively in the way I describe it is bacteria that make your tummy tumble. So cholera, Shigella, some of the other things, norovirus, not a bacterium, but things that really you want to make sure your meat is as cooked or as well prepared as possible so that you don't get them. And in that space, I was also in the vaccine development space, thinking about how do we create vaccines that work in young children and only need one dose, so you're not requiring multiple visits to the clinic if you're in rural Bangladesh or in rural Nigeria, like where I grew up. And then even before then, working, as you mentioned earlier, Tim, in diseases like malaria and tuberculosis, where these are things where I can give you some shocking statistics about the number of people in the world who carry these diseases and the economic burden they have, but also how much it can really make a difference on a country or international scale if you're able to get valuable therapeutics and vaccines against them. So really sticking within things that, like I mentioned earlier, come into your body and make you really sick, but trying to address them in different ways from a research perspective and now in a more business development perspective side.
are you employed by any particular pharmaceutical, or you're as part of Harvard, how is your research given to the pharmaceuticals, or how has that transition happened? Because I imagine there would be a lot of paperwork involved with it.
No, got it. This is a fantastic question. So I can tell you a little bit more about my transition through the PhD, where I started with a vaccine for cholera that we really didn't know how it worked, and we really didn't know whether or not it was the best vaccine available. So I spent 5.5 years working with different animal models, different things in the lab at the bench, trying to really interrogate what makes this vaccine so good. And at the end of the day, you release that to the CDC or the FDA and you say, okay, here is all the data we've gathered that proves that this vaccine is the best vaccine out there. It will provide protection against all the cholera in the world, and we have all this data to prove it. What I do now is on the other side of that, where a company takes that idea and starts to distribute it. They start to work with specific pharmaceuticals, or they start to work with major biotechs to really take it to scale. I think Kevin can speak a little bit more to this when he said that like some of the mouse work he's doing takes a really long time, and you can't really do that at scale or to a certain level in a sort of academic environment. So what I'm helping now is a bunch of different pharmaceuticals take these sorts of ideas and then like bring them to people who need them. We work with pharmaceuticals that are doing things in rare diseases or very specific cases, but also, like I mentioned with COVID, we're trying to figure out, okay, what does the next few months look like? And how do we just like prepare ourselves against that?
Bolu, can you tell the room about where things stand vis-a-vis clinical trials for that vaccine work? Because when you defended, I remember that I think that data had just gone into clinical trials. That was really cool. Were there any updates since then?
Yeah, so the vaccine that I worked on is currently being trialed out in subset populations in the Boston area. And what's really cool about these sorts of vaccines is that you can measure two things. What is the immune response of the vaccine when it goes into a human being, and also how protective is that immune response? And that's something that you really can't do with a bunch of other sorts of diseases. So it's been fun watching that project grow and blossom out of something that if you'd asked me 10 years ago, would I have been working on something like this? I wouldn't have known how to answer that, to, oh, it's actually making a difference in people's lives. And in the next 10 years, we'll be going into children under the age of 5 who really need protection against cholera in certain parts of the world.
You're testing it on the East Coast. There's who are the people who you're testing the vaccine on? I would think that there's no cholera back there.
There isn't, and that's a fantastic question. We have different test sites, the East Coast being, well, there's 2 levels to these sorts of studies. The first level is it safe in human beings? We do a lot of work in animal models, and we validate and we show that it is biosafe, but you also want to make sure that in a controlled setting, things are happening as you expect them to. And then you go to the field sites. And so we have some collaborations, and these are projects I'm still working on in collaboration with Harvard and the other hospitals in Boston, in Nigeria, in Sub-Saharan Africa, in South Asia, in East Asia, where we're really trying to make sure that these vaccines are providing the protection we want where the disease is found. Because the things that people are exposed to there are very different from what you're seeing in Boston, New York, and Philly.
Sure.
And Bolu answered the question pretty beautifully. The only thing I'll add in terms of, you know, how we work with pharmaceuticals or therapeutic opportunities is very frequently, especially depending on where you are, but like Harvard being in Boston and Cambridge, there are so many biotechs here.
Mm-hmm.
And so many startup companies and therapeutic companies. So my professor actually just told us that he had a company. And this is something that really happens frequently at pretty much every university, but especially ones in Boston or, you know, the Bay Area, San Francisco universities.
San Diego.
They love to spin out San Diego. They love to spin out companies based on their research. And, you know, it's not really possible for a professor to run a lab and run a company. And that's why they have very strict rules about it, but they usually can help found a new company and basically streamline it so that the research that's being done in the academic lab ends up being the basis for a new company's new drugs that go to trial. And so that's something else that you see pretty frequently. And I will also add, as Bolu kind of hinted at, this isn't just in the biotech hubs. It goes down to places like San Diego, which 30 years ago was not a biotech hub at all. And then over the years, a lot of companies started opening up, and there's no reason to think that can't happen in areas like the Central Valley as well, where you just start seeing, these booms of biotechs.
Like, Kevin, would you mind talking a little bit about the research you're doing now with cancer?
Yeah. So my lab is essentially focused around the fact that when someone gets a tumor and they get that tumor biopsied, the amount of information that we get from that now versus even 10 years ago is worlds different. And what I mean by that is our lab studies cancer genetics. What are the genetic mutations that we find in a patient's biopsy? Because that actually has huge implications on whether that patient will respond to the chemotherapy and survive. If a patient has this one specific mutation, they might be a responder. If a patient has this other type of mutation, unfortunately, they're not eligible for this breaking treatment because we know it won't work in them, which is great because it saves that patient a lot of time and money and doesn't get their hopes up. Conversely, if a patient is, looking for hope, finding the right mutation that works for them, finding the right medicine can give them hope in a very kind of bleak setting. So one example I like to give is I had a chance to shadow an oncologist early on in graduate school, and we were looking over the patient's data together. He had just gotten back the sequencing data, the DNA sequencing on her biopsy, and he was excited because he said, "Oh, this woman has Lynch syndrome, which means she has a genetic mutation in her tumor. Actually, it was an inherited mutation, but it led to the tumor, which is unfortunate. But the really fortunate part is that it made this patient one of like the 10%, the 5 or 10% of people who would actually respond to some of the like most effective immunotherapies we have right now." This kind of conversation didn't exist 10 years ago. I had a family member who had a brain tumor in 2008, and I tried looking into it and just seeing if I could understand his case with the perspective I have now. That data doesn't exist. And so this is the kind of thing that's really new, really exciting, and that's what my lab studies. We use mice to try and test out different mutations and see the effects that they have on cancer, and potentially see how that relates to therapy effectiveness. It's a really exciting time, not just for cancer, but biomedicine in general, in case you couldn't tell from all the cool things Bol is able to do with infectious diseases.
So pharmacogenomics is the way of the future.
I'd certainly like to think so, but I'm biased.
Okay.
I mean, a point I'll add is that, Kevin, how much of this have you learned as you've been doing it? Like being able to pick up these skills and the background you had versus what you know now, you can really like get stuck in and really learn as you go, right?
Oh, absolutely. The level of on-the-job training that both Bolu and I experience is pretty wild. And that's why PhDs tend to take a bit longer, or a lot of graduate degrees. You're adding, several years onto schooling in addition to college. But I mean, it really teaches you what it means to become an expert in something. And there's really nothing other than taking the time and just doing it and picking up these skills day by day, as Bolu mentioned.
Well, while we're on the subject, what would a typical day look like for either of you as far as in your research? What do? You get up in the morning, have a cup of coffee, go.
I'll start. I probably stick more to tea, but yeah, totally. I'll start off since I'm still in academics, and then Bolu can also discuss his academics and mention what it's like outside of that in consulting in the business world. But yeah, I think It varies a lot because I kind of set my own calendar. And so, some days, some weeks, you know what? I'm really tired. I'm really busy. I'm going to start my day at like 10 a.m. Now, conversely, that means I'm going to leave the lab much later at like 7 p.m. or 8 p.m. even. But that's kind of what it boils down to is I have a flexible schedule. I have a list. Sometimes, especially if you're just starting graduate school, it'll be just like a bullet point list of like, here's the things I need to do. And then as you start to get more and more advanced, you get more feel for what an experiment looks like, what the steps are. And so for the point I'm at now, you just have like a very thorough flow chart essentially for like, okay, I need to hit this part, bing, bang, boom, just do all these parts to hit my full experiment before the end of the week. The actual experiment varies a lot. Some days I'll be in the mouse room for 3 or 4 hours, studying the tumors in the mice that my lab uses. Some days I'll be at my bench, mixing chemicals to create a reaction that will tell me something, measuring levels of a protein in my sample, for example. Some days I'll be in a tissue culture hood working with actual like tumors in a dish, essentially. So that was my day today. I spent the entire day working in tissue culture hood. But those cells that I worked with today, they don't need my attention for the next 48 hours. So tomorrow I'm actually focusing on an entirely different part of the lab. I'm going to be working on a different experiment at my bench. So, I think Bolu will touch on this, but the point is every day is different. Obviously there are some parts that can be a bit monotonous, especially if you have an experiment that's like very repetitive where you have to do the same kind of thing over and over. But I think for the most part, because there are so many different types of experiments we do from day to day, it means that the bench work we do varies a lot. But I should also mention, I need to actually make a plan for what I'm going to do at my bench. I need to analyze data. So while I do spend a lot of time at my bench, I do also spend a decent amount of time at my desk on my laptop, figuring out what my data looks like, what it says. So it's kind of a hybrid thing. It's not exactly an office job. Even though I do have office space with office supplies, I'm frequently on my feet in a way that you often wouldn't see in most jobs. And so I think that variety is pretty nice.
How many people do you work with in your lab?
So my lab has about 20 people, but I think the thing that is really interesting about working in research, whether you're studying, pharmacology or cancer or infectious diseases, is you'll frequently collaborate with people outside of your lab. So I, 3 days ago, went over to another lab in another building right next to us and worked with a postdoc in that lab and talked with him about his paper and trying to replicate one of his experiments. And so, any given student, any given year, they might be working with not just, several people, but dozens of people really. And I think Bolu can speak to this. He collaborated with so many people during his PhD.
No, I really like what you said, Kevin, about like planning things and getting them ready for the week and thinking about like week-long increments, because the cells you work with divide on a rate of what, every few hours versus days, where the ones I worked with divide 20 minutes and ready to go. And so that was definitely something I wasn't expecting going into well, I knew about it, but I didn't know how to really think about what a day looks like when you have X number of things on the burner and you want to make sure these experiments get done. But a day for me would look like Kevin said, like you start whenever you really are ready to go into the lab, ready to stay at the bench and use the pipettes and get those experiments done, ready to work with the bacteria and do all the cloning and all the things that are really cool theoretically, and then also really cool in practice when you realize the kinds of the things you can do in biological systems. One other thing that I'll touch on is that the graduate process or the ability to go through the PhD really lets you dig into teaching and mentoring. And so a fair amount of my day was spent teaching classes to other students and grad students and master students, a lot of it in biochemistry. And so those class times are a little bit less flexible than my regular experimental timeline, but beyond that, a very similar sort of work days. And now I work in a space where a lot of it is defined by what the biotech is looking for. Do they want daily meetings? Do they want weekly check-ins? Do they want bi-weekly check-ins? And what's the data that they're looking for?
Questions? I don't want to dominate the conversation, but if you had anything in particular you want to hear from either of the speakers, how effective is the COVID vaccine?
Extremely, especially the more recent combinations that reflect what circulating COVID looks like. I think effective is I think you can think of effective in two ways. It can either be what does an experience of COVID looks like versus severe COVID, which we all don't want to have. And these vaccines will very much close to 100% protect against severe COVID. So it's really just trying to minimize spread and minimize risk associated with that.
Got it.
Yeah, and I think, oh, sorry, go ahead.
I think we have another question too.
Most of the disease, you know, that when you have vaccine, can you hear me?
Oh, a little bit.
I'll walk over here.
Thank you.
Well, Tim will repeat the question as well.
Thank you. Okay. We know that most of the disease, when you get the vaccine, you will be like cured for that disease, malaria and other like chickenpox and so on. Why COVID when even you get the vaccine, you get again infected or you get COVID and there's a dosage 1, dosage 2, dosage 3, and we've heard that it's going to be an annual vaccine shot. So why this disease? Is that because it's new or undiscoverable or?
Bolu, may I take this one?
Please go ahead.
So that's a great question. And the answer is it's not just COVID, but we only started to appreciate this like right before COVID hit. And the reason that, you know, this makes me laugh a little bit is because I can remember this. I actually remember I remember the exact chart because I thought, wow, this is really interesting. I was sitting at my desk, you know, reading through papers. That's what we do most of the time. We're trying to stay up to date on stuff. And it must have been like 2018, 2019, right? Right before the pandemic. And this chart was showing that, you know, the length of protection from a vaccine can vary a lot. For some of them, like the ones that you've mentioned, their lifetime, you know, chickenpox, for example, being, I think, a good example. So some protection lasts a lifetime, but it varies dramatically. And for some of them, and I can't remember which ones, some vaccines have much shorter, longevity. And that's something that we really didn't appreciate as much until right before COVID hit. And unfortunately, I don't think we had enough time for that to really kind of like circulate into how the general public thinks about vaccines. And so by the time that COVID hit and it was like, oh, wow, vaccines don't last forever, that was a surprise for most people because we only recently started to understand, okay, vaccines are really useful, but some of them need updates more often than others. Bolu, is there anything you want to add to that? You're more expert in this.
No, you hit all the major points. I think the other part is that a lot of the strategies that we're doing now are to remain ahead of the game. Like we want to create a situation where there is nothing on the horizon. And so if we're being overprotective, that's a better strategy than trying to react to things. And so a lot of these things that we're recommending or putting in place are to make sure that worst case scenarios don't even happen to begin with. And that's where a lot of the, like you mentioned, yearly dosing or things comes in, because we want to be in the best place possible so we don't have another situation like we had a few years ago.
Like the flu vaccine, we'll get a different vaccine every year. I think the concern some people had was the use of the mRNA with the COVID vaccine. Can you talk about that a little bit and why that would be? I don't want to say a concern, but I know that was an issue that some people had with it. Is it was a new type of vaccine.
So I can speak to a little bit about mRNA, Kevin, if that's okay.
Please go ahead.
I think that when a new drug comes out, we all want to make sure that we understand how it works. And the mRNA vaccines were not something that we had familiarity with. That being said, they are very effective. And even though they represented a new technology, they went through the same validation process that every other kind of vaccine goes through. They went through the preclinical models, which I have expertise in. They went through the clinical trials, and clinical just meaning in human trials that a lot of people have expertise in, and they went through the scale-up processes. But what was really useful in this past couple of years was that we learned that we could really make that system faster, but still keep the same number of safety thresholds or safety requirements in place. So they are a new technology, but they're incredibly efficacious. And what's really useful now is thinking about beyond COVID, what are the other things that we can begin to address with mRNA vaccines in a way that we had no idea we could before? mRNA it's tied to what we are as human beings. It's in all of our cells. It's something that we're very familiar with, and our bodies know how to develop immunity to mRNA-based vaccines. And so now it's like the world is our oyster. We get to think about what diseases, what cancers, what things could we not think about treating or preventing before, because we didn't know we could do them with mRNA vaccines. But we've seen how COVID vaccines have been really, really good these past few years. We can now venture into those spaces.
In a way, COVID opened the door for mRNA vaccine production in a way that it might not have come onto play as soon as it did.
Mm-hmm.
Definitely not. And there's a lot of excitement about that technology to be applied in the cancer field. People have been talking about cancer vaccines for a while. It's been one of the very kind of hot topics in the field, but it kind of goes in stops and starts because I think a lot of the trials are generally not super successful. And so one thing that might revolutionize that is mRNA technologies. And one thing I will just add to what Bolu said, scientists, one of the things that scientists have always been incredibly good at, even before modern times really, is manipulating biology to completely cancel out any risk while we use something that would have previously been risky. And so, for example, every week, there's someone in my lab using bacteria. It's E. coli. This is the same, in theory, this is the same E. coli that if that gets on your lettuce in the farmer's market in Visalia, there's a recall or there's a huge outbreak. The reason none of us are worried in the lab is because we've manipulated that bacteria to be non-pathogenic. In other words, it can't, infect and colonize. Bolu, how long has non-pathogenic E. coli been around? It's decades old, right?
It's decades old, and it's so interesting that you bring that up because the strain I'm pretty sure you work with came from someone in a California hospital where they realized, oh, we could just turn this into a lab strain and make it do all the things you want to do, as Kevin mentioned, by de-risking it and making it safe to work around people. So you can really take something that was dangerous and strip it down and take away the things that are pathogenic.
And this is something we have done, again, for over half a century. We did it with bacteria. We do it with viruses. I regularly work with viruses that used to be from the same class as HIV. I'm not scared at all. And there's no reason to be because we have manipulated them so they can't actually reproduce in a body, essentially. You use them for infection once in a petri dish, you're done, those cells are dead, the virus is dead. So this is kind of something that scientists have been able to do for a really long time. And although mRNA is a new, technology, it's the same exact concept we've been using for a while. We take something and we make sure that we use it in a way where it can't replicate, it can't make permanent changes. And so the mRNA will stimulate your immune system, but it's not DNA. It's not going to change your DNA. It's the kind of thing that science has been really good at for a while. And that's why I bring up the bacteria and the viruses as examples of ways that we've done that for, almost a century.
Well, Balu and Kevin, I think we're out of time for our first session. I wanted to thank you guys, and I appreciate everything you were able to share with us tonight. And I know our folks here are going to move on to session 2, hopefully. We have some other eager individuals who want to come, and want to thank you both.
Of course.
Thank you.
Of course. Thank you for your questions.
Thank
Now,
you.
Tim, I think we volunteered to do sessions 1 and 2, right? So we'll be sticking around for session 2. Am I remembering that correct?
Yeah.
Yes, you will.
Yeah,
Or there's going to be someone that's upset with you.
Yeah.
You'll have to answer, during the holidays.
I think you might be allowed a potty break now, though, if you need it. Do we start at 7?
So the next session starts at 6:55.
So 4 minutes. You better run.
Run, Kevin, run.
Sounds great. It's good to see you, Paige.
Good to see you. Thank you for doing this, Kevin.
For you, anytime.
Did I
One
ever
minute.
tell you this is my favorite son-in-law?
That's
My only son-in-law.
Uh,
But,
yeah.
I'm recording both these sessions with them so that if you decide which one you like, and I can
Uh-huh.
clip it and edit it and send it, and you can show it to your class if you want.
Oh, okay.
Okay.
Very good. Hi, good evening. How was your first session? Did you go to a first session? Yeah. Who did you hear? Oh, okay. Which was? Oh, fantastic. Did you go to us? Oh, the same? Excellent. Great. We'll start in a couple minutes there, giving them a little bit of a break. So you look like you're a high school student. Where do you go? And what are you interested in the health field? Neurology. Okay, excellent, excellent. This young man is in what grade? 8th grade? Where at? La Jolla. So you're going to come here and be part of the Health Science Academy. I'm going to recruit you if I can. I'm the lead teacher for the Health Science Academy. My name's Tim Parada. And this is a great opportunity for you to hear from some researchers in Harvard, which is nice. I try to recruit you, but you're already at Redwood. So yeah, we have a really good program here. I'm happy to tell you more about it, but that's something you're interested in. Is that the field you want to go into? You're thinking? Making up your mind? Yeah. Okay, good. Good. Hello. Okay, well, I'm used to hearing bells ring, but we're not going to ring bells tonight, but we are ready to start a second session. Kevin and Baloo, I'm going to have you introduce. We've got several students here along with their parents, and they're interested to hear what you folks do. So I'm going to turn it over to you and let you do your introductions.
Hey everyone, my name's Kevin. I'm a senior PhD student, which effectively means I've been studying in my area of cancer biology for several years now, but I should backtrack and explain how I got here. So I grew up in Southern California, San Diego originally, and back in high school, I enjoyed biology classes. I thought they were neat. Wasn't sure if I wanted to do that for the rest of my life, kind of a big commitment. So I went to university, got myself a bachelor's to see how I liked science and biology. I went to a UC, just a little north of San Diego, UC Santa Barbara. They had a really strong biology program. I was interested in biology. I decided to check it out. And, you know, I took it day by day. Every day, I asked myself, do I like this? Do I want to switch majors? Econ? Psych? Nah. I ended up sticking with bio, really loved it. I focused on biochemistry because I did really fall in love with chemistry in college. I just really loved seeing things on the nitty-gritty scale that biochemistry lets you see and study. And at that point, that was my first time that I got involved in, you know, research and working in a research lab. Nowadays, I think a lot of high schools are giving students the chance to do that. I know Tim is running several different kind of recruitment campaigns for these fantastic programs and academies that give you research experience. But for me, my first time doing that was freshman year of college, working in a neuroscience lab that was studying proteins involved in Alzheimer's disease. And it really opened my eyes because studying biology in a classroom is one thing. Working in a research lab and actually trying to make biology tell you about itself, that was another experience. And I enjoyed it enough that by the time I graduated, I wanted to do it more on a higher level. And I applied to PhD programs where I could run my own projects and hopefully create my own discoveries. And so I ended up coming over here where I am now to Harvard University in Boston. And I say Boston, not Cambridge, because I'm at the medical school because my program, and the same one Bolu was at previously before he graduated, was the Biological and Biomedical Sciences PhD program. So, you know, I wanted to do research, but I also wanted to focus on people and diseases that they faced. And that's where I am now. My area of focus is cancer biology. I can expand on that a little bit later and say what that involves, but I focus on cancer. I use mouse models. I work with these really small, cute mice, and I see what happens when they get cancer, which sounds brutal. Some days it is, but it does, you know, help us make decisions about what can save people's lives. So that's how I justify it anyways. Bolu?
No, so Kevin and I did meet at Harvard, and I graduated with my PhD last May. And a lot of the stuff we did was very similar in terms of our day-to-day, how we structured them, how we designed experiments, and how we understood the data. But we had very different backgrounds coming into biomedical science. My first lab experience wasn't until my junior year of my bachelor's degree. And mostly I came into thinking about what I study, which was infectious diseases, from a lot of the experiences I'd had. So I was born and raised in Nigeria and really saw how diseases like malaria, tuberculosis, cholera really caused a major social and human effect. And so my bachelor's degree is in anthropology. And a lot of that was spent thinking about what happens to a patient when they go into a hospital. How are they being treated? How are they understanding their disease? And through that, I realized that I wanted to learn more about these diseases, how they cause the problems that we saw, and also how we could help to prevent them. And so from my first research lab experience, I realized that there was a way I could not only think about these diseases, I could really get plugged into them and start finding out answers myself, like really start making discoveries and contributing to these processes that I didn't know I had any access to before. So I decided to do a PhD. Kevin and I met at Harvard. I focused, whereas he was looking a bit more at things that we consider intrinsic, the cancers where cells go haywire, I focused a bit more on what happens when a pathogen tries to infect your body and take over the system. And so I spent most of my time working on understanding these pathogens, but also developing vaccines against them, being able to prevent them creating disease in animal models, and as my work finished up last May, in human beings as well. And currently I'm continuing that sort of work, thinking about how we can help businesses bring these vaccines or these therapeutics to patients in the clinic.
Great. At this point also, if you have any questions at any time, I'm sure they're happy to answer. If you think of something you'd like to know, feel free to ask. I'll give you the microphone. You can talk to either one of them. Last session, you folks were talking a little bit about the current work that you're doing now. I don't know if either you want to take a moment to talk about what it is you're doing and what you're studying right now or working towards.
Bolu, please.
Yeah, no, so a lot of the work that I did at Harvard, and Kevin made a point to mention that we were at Harvard Medical School, which is really important because you see not only what you're doing in the lab with the mice and the other animal models and the bacteria and viruses you're working with, but you see how that influences patients in the clinic. You're right next to the hospitals, you're right next to places where you're seeing these findings have impact. And so that kind of inspired me to want to take all the things that I'd learned from my undergrad, from my high school, from my PhD, and really apply them outside of the academic space. So currently my day-to-day looks a little bit different than what it did a few months ago. I spend a little bit less time at the bench now. I work with fewer animal models, but at the end of the day, Kevin and I both think about science in a very similar way, which is, what is the patient experiencing? How can we make that better? And how can we bring things to them that will increase or make the outlook a bit brighter? He's coming at it from a very, and you can correct me if I'm wrong, Kevin, from a more academic and research-oriented goal, and I'm coming at it now from more, how can we get this to the patient? But those are both the same goal at the end of the day. We're both trying to make things better for people from our different perspectives.
Yeah, I think, Bolu, you hit the nail on the head. Something we both studied, also in terms of like nitty-gritty specifics, was the immune system, which was really fun because Bolu and I, completely different fields, right? Cancer biology, infectious diseases. But we both ended up talking a lot about our immune systems, not like just ours. And obviously we would complain to each other if we got sick, but we talked about humans' immune systems and what that looks like in terms of fighting off, cancer or infectious diseases in Bolu's case. And that's what my project is on. It's what gets me out of bed in the morning. It's just the idea of every day, right now, as we speak, your white blood cells are killing a cell in your body that was going to become a cancer. Your immune system is really good at this, and that's what I study. Is how can we better understand that, because that's something that's currently being used in clinics. 5 years ago, it revolutionized the world of cancer therapy because there were patients who had metastatic diseases, tumors throughout their entire body, and that was effectively a death sentence. But suddenly we had these drugs, these immunotherapies that would activate their immune system and the cancer would melt away. And that's not slang. That's what the doctors would actually say, is they would say it melted the tumors. It just completely got rid of these metastatic cancers. And there's a lot we still need to learn about how that works. I think most patients, that doesn't work for them. It's only some patients that this is really effective for. And so I try to understand why that is and what that looks like.
Questions?
I could go on for ages.
Oh,
In fact, I've been going at it for 6 years now. This is my 6th year of PhD work. So I'll stop it there. But if any questions come up, I'm happy to answer.
could you expand a little bit then about how it works for some people and it doesn't work for others? Because I know that's an interesting topic and the future of pharmaceuticals, really.
Sure, absolutely. I mean, that's a Nobel Prize winning question, first of all. So if I answer that question successfully, you know, I'll be a very happy person. But you know, we have some insights. So I think in cancer, a big reason why immunotherapies might not work is because of the tumor, specifically where it comes from. So there are certain types of cancer that are really good for immunotherapies. For example, if you have skin cancer, the most aggressive form of skin cancer, which is metastatic melanoma, that is super responsive to immunotherapies. A much higher percentage of patients will respond to that. My lab focuses on a different type of cancer, and that's colon cancer, cancer of the large intestine. This is super common, especially in adults. You know, once you hit 30 or 40, it's recommended that you regularly get colonoscopies, and that's because this is a really common type of cancer. Only about 5 to 10% of colon cancer patients even have a chance of responding to, you know, these immunotherapies that have revolutionized melanoma patients' lives. So at first, it boiled down to just what type of tumor you had. What my lab focuses on is the actual genetics. And this is something we've started to appreciate, is the genetics has a really big effect. So the 5% of colon cancer patients who do respond, genetically, they have something in common with the melanoma patients who respond. So it's actually led to this huge breakthrough where we've started to, the FDA, the Food and Drug Administration, has started to approve drugs not just based on the tumor type, which is how they used to do it for decades. They've started to approve it based on the genetics of your cancer. If your cancer has this specific genetic profile, you are approved for this drug. And that's what we're trying to study. It's really new. This was not a thing that existed 5-10 years ago. And so it's just a really exciting field. There are a lot of questions to ask. It's hard. These experiments take a lot of time and effort, and they're very easy to mess up or design poorly. But at the end of the day, it's really exciting work, and there's so much to still discover.
I will add a minor plug about the Nobel Prize that you mentioned earlier, which is that so much of this work that we do has unintended consequences, which is that you discover things along the way. As we're learning more about the human genetics that define what makes a cancer be able to spread to certain places and others can't, or what makes one person more susceptible to certain diseases than another person, we're learning more about what it means to be genetically human and what it means that our DNA is contributing. And those are things that even if the experiment goes bad or you don't get the data you were looking for, there's so much learning across the way. I think something that like kept me going through my PhD and continues now is just how much potential for, there are things you're discovering that no one else in the world knows. And that's something that you get to own. You get to say that I discovered this, this is my contribution. And it's a really wonderful way to engage with learning.
Although you want to talk a little bit about what your position is and how it evolved to where you are now and what you're doing for pharmaceuticals?
Sure. So I started in the vaccine space. I started thinking about diseases that were very important to me, malaria, tuberculosis, HIV, cholera. And then I spent 5.5 years developing a cholera vaccine at Harvard. And at the end of my time at Harvard, we were able to get that vaccine out to the clinic, out for the testing, out to make sure that it really does what we need it to do and will provide the protection that we want it to do in the parts of the world where it's most needed. So that was where I exited that project and got my PhD. And like I mentioned, learned a ton of things along the way that I wasn't expecting to learn. But now what I'm doing is thinking about science from a different perspective, which is how can I support businesses who have all these different compounds or drugs or therapies, get them to the clinic, get them to the patients. And a lot of that is using my knowledge of biology to be able to make a judgment call, like this is a good vaccine, this is a good product, I've seen the data, I understand what's going on. But the other part of it is being able to recognize whether or not it's sustainable, whether or not it's something that requires funding from certain sources or others, and really tie that all in together to create a business case. And that's been really fun for the past few months, like seeing something that came out of a lab, a research lab, one like Kevin's, that's able to understand what's going on the human side to better explain these cancers, and then take that right through to the patient who needs this, who's able to say, ah, yes, this is going to melt the tumor away. That's where I am currently.
Any questions, folks? From discovery to distribution, what's the timeline like?
Depends what you're asking about. From discovery to distribution, it can look as short as a few months, as we've seen most recently. Typically, it takes in the order of 2 to 5 years, 10 years on the longer scale.
And FDA approval is part of that process?
Yeah, so a lot of the benchmarks that we use to make sure that things are considered safe, and not even safe, but beneficial, remain consistent. There were no corners being cut or edges being shaved off. It's all the same.
So what advice could you give to our students who are here now, either looking to go into a medical career or in the health field, as far as what should they be focusing on in secondary education?
I would say question yourself constantly. Like, don't be afraid to ask yourself the hard question of, do I like this? You know, Bolu and I can talk about all the things that really get us jazzed and excited about science, but at the end of the day, it's hard. It's hard work. It's a job. And it's good to do something that you actually care about, that matters to you, and that you're enjoying. And so, for example, in college, every day I was asking myself, do I like this? Should I switch to a different major? Or for example, Bolu mentioned he started as an anthropology major. Bolu, did you interrogate yourself sometimes about, what direction you wanted to take it?
100%. And I think the only thing I'd add on to your solution is to find people who make the journey worthwhile. I think that I'm incredibly grateful to have someone like Kevin in my life, but I can also look back at the points in high school, in college, where it was either friends or people in classes or mentors. I will just stress that finding people who have been through it before, can point out the mistakes that you don't need to make so you can make your own mistakes and learn from those is so, so important. I would not be here, I would not have my doctorate or the things that I'm proud of without I can point them out person by person. And that, I think, is such a critical part, that once you enter a new environment, especially one that you feel uncomfortable and you're questioning and finding out if you want to do that day to day, you just have someone who's a point person and seek them out.
Also,
I'll go ahead, Kevin.
Something more specific, but just that Bolu really brought up. When it comes to mentors, go to office hours.
Yes.
It's like the single most useful thing I did in all of college, is just walking into the office of my teachers and saying, hey, I'm confused about something. Can we talk about it? That's what your teachers are there for. That's what they love to do. And office hours, especially at the university I was at the time, are criminally underutilized. Most students do not use them as much as they should, which, to your benefit, means you get a lot of one-on-one time with your teachers, who are usually some really cool, really smart people.
What was the most difficult part for you transitioning from high school to college?
I'll take a stab at this.
Please.
I didn't know how to ask for help. I spent a lot of high school thinking that I knew what I was doing and I knew the trajectory I wanted, and I knew that I wanted to go into anthropology and really just like dig deep into these things that I'd read about. And when things started getting tough, and when I started realizing how much I wanted to study the diseases and not just the impacts they had, I didn't know how to make that transition into organic chemistry or the biochemistry or the math or the statistics or the things that really make up a lot of those degrees. And so that transition of being able to go to office hours and say, hey, I don't really think I understood that lecture. Can you break this down for me? And then realizing that the professor wanted to do that because it's something they signed up for was a mental shift I had to make. And then the only other thing I can point to right now on the spot is taking notes, just writing everything down and keeping some record of it to be able to go back to during like exam revision time or something like that.
I think Bolu brought up a great point. The only thing I'll add is the biggest thing for me in terms of transition is the time spent in class. And this is such a huge difference from anything you've ever experienced before college, right? How often are you guys in class? Like raise your hand if you spend 8 hours in class every day. Is it 8 hours, 9 hours? Yeah.
Well, 8:30 to 3:30 with a 40-minute lunch. So you figure 7 hours maybe a day.
Yeah, 7, 8 hours. So multiply that by 5, you're spending 35 to 40 hours a week in class. When you get to college, each class you take will usually be like a 50-minute lecture. And then maybe there will be like a discussion section once a week. That's an hour. And you'll have 3 lectures a week. So do the math, you end up with about 4 or 5 hours of class the entire week, compared to 40 that you were spending in high school and middle school. That's a huge adjustment. And I think when you first get to college, you're like, wow, I have so much free time. You don't. That's a lie. There's still studying that you have to do, but now you have to do it on your own, and not necessarily like you're by yourself. Bolu mentioned there are great ways you can ask for help, but you have to learn how to do that. You have to go to office hours. You have to talk to people. It's not just a teacher who's always going to be there. It's you pushing your own education. And that's a big adjustment. That was something that threw me off at first. And it quickly made me realize I need to get good at scheduling. And that helped me a lot when I first transitioned to college.
Did you attend study groups or work with other students when you had difficulty in classes?
Oh, absolutely. I mean, like Bolu mentioned, it makes the difference between, succeeding and failing is being able to lean on friends and study together and help each other understand things.
I will add that's how Kevin and I met during a particularly stressful computational workshop that we both needed to understand for our respective studies, but studying together and really being able to support each other, there's some things that having someone explain it to you really just makes a huge difference, as opposed to trying to read it from a textbook. That's a big deal.
Well, I know as researchers, you talked about occasionally you'll make mistakes in the lab or in an experiment. Well, what mistakes would you say you made in your career as far as college education goes? Was there anything that you would advise students to avoid or to help them?
That's a good question.
Put you on the spot.
I will say probably just, not valuing sleep as much as I should have. I think, college is an exciting time. There's a lot going on. There's a lot of stuff you want to do at once. Classes, socializing, all that. I would have slept more, and given my body a little more of a break. You know, you're young and you think you're invincible. And then one day you're like, oh man, I'm actually really tired because I didn't take it easy this week. So building habits of treating your body better is something I probably would have wanted to start a little earlier than I did.
Something you brought up earlier, Tim, was how important it was for students to get on-the-job experience and really understand what things look like on a day-to-day basis, like X 100-hour internships, and really know what it looks like on the ground. There's a lot that I learned in the library. There's a lot that I learned in the classes and the study groups. There's a lot more that I wish I had gotten from the day-to-day doing extracurriculars, engaging with volunteer opportunities, really finding out what resources the program I was enrolled in had to offer, and then doing those to make sure that I was getting an understanding of what healthcare and infectious disease look like, not only from the textbooks, but from the day-to-day research perspective.
Did either of you spend any time in the hospital or working with patients as well?
I did not do that in college. It wasn't until grad school that I had a chance to actually shadow a doctor and be in the room with patients. So it's a huge privilege. If you have a chance to do that as early as high school, you know, and you have even the slightest interest in medicine or research, it's a huge privilege. And I definitely recommend taking the opportunity.
I did not. I had friends who were pre-med or pre-PA or pre-nursing who spent time in hospice, and they would come back with stories about how there's only so much you can learn about glycolysis until you see what a major impact that has on someone's life. And that really inspired me to learn, you know, more about glycolysis. And so I switched from my anthropology background to a more STEM-based field.
Any questions? Yes. Hold on. Let me get you the microphone. I'll be like
Hi. For both of you, what was like the most challenging or the hardest like research you did, and what kept you motivated? Like sometimes we feel like we're like want to give up, because I know like for these researches maybe take years and years, and sometimes you don't see like results right away. You need to be patient. How did you keep yourself motivated and like pushing yourself through it and didn't feel like giving up? Thank you.
That is a phenomenal question. Bolu, do you want to go first or should I?
You go first.
Okay. I'm going to focus specifically on, I think, what you were asking about, which is during research, because, you know, the ways to stay motivated during college are very different versus like just doing research. I think the biggest piece of advice I ever got was you need to be able to separate the results from the person. You are going to fail. Like that is a guaranteed inevitability in research, right? That's the whole thing. It's research. You're trying things over and over. You're going to have experiments fail. You're going to have things go wrong. And you need to be able to say, okay, my experiments and my data are not reflecting who I am as a person. That's just how research goes. And it doesn't mean I'm a bad scientist. It doesn't mean I'm wasting my time. That was super helpful to hear. And it really helped me when I was struggling and things were failing. So I think that's the biggest piece of advice I would give. Bolu?
I think one of the things about doing research, especially during the past few years, thinking about infectious diseases, is getting so caught up in your own research lane that, or for me, I forgot where the work I was doing attached to other people's work. And so being able to take a step back and say, I am carving out this new area. No one in the world knows this information, and I'm going to present it to them in a way that they can understand. It didn't come out of nowhere. And so being able to like take myself out of that stressful environment and see where it came from, where the influences were, and how everything is sort of connected to each other. Kevin mentioned earlier that we both study the immune system. There's a lot that I transmitted to my vaccine work, just talking with Kevin, being like, okay, this is how the immune system works in other cases. How can I think about it in this specific case? And how can I like get that to overcome this milestone? And that's something that really just helped me get through those particularly charged or stressful moments.
I think she also wanted to know what sort of things keep you motivated to go into the lab every day and to spend 6 months waiting for the results of one experiment and so on.
Both.
A lot of the work that I did was motivated by what I was familiar with in the disease space. At the end of the day, or at the end of a research project, or at the end of a chapter of my thesis, I would think about what impact this is going to have on the world. And that, above all, remained a motivating factor. These are discoveries, these are things that you get to lay claim to, but also release out into the world that will have an impact. There are children under the age of 5. We use 5 as a benchmark because that's something that pediatricians have decided, and we just use that for our vaccine work. But this is going to make a difference in their lives. And so thinking about the broader scope of the broader impact of this one experiment or this one chapter, that really helped get like through motivate things throughout the day.
I think the only thing I'll add to that is just the research we do, the experiments that we try and fail at, we do them because they're part of a project that we created. You know, we're the people who thought of the question. We're the people who said, what happens if you do this? And that's because we're interested in it. I'm really interested in this one protein right now. That's what I'm focusing on. That's what all my experiments focus on, because I think this one protein really affects how the immune system interacts with cancer. And so, you know, that's like my personal idea. That's like it's a sense of ownership that I don't know if I've ever really had over anything. It's not like you really own a lot of the stuff you cover in classes, except for like maybe when you're writing like an essay and creating your own ideas there. So I think that's the big thing for me, is just feeling a bit of a sense of ownership of like, these are my questions. These are the things I'm wondering about. And these, you know, these questions will get answered. Even if my experiments are failing right now, there's going to be a way to answer them sooner or later.
So right now your research is being, oh, did you have a question? No, go ahead. I was just going to ask them if they ever thought about going into private industry, but that can hold off. What were you going to ask?
My question is, you guys are in Harvard, right? It's an Ivy League school. What advice and recommendation do you have for our children that are going to go to high school? And what sets them apart from other students that'll help them get into those Ivy League schools?
Great.
That's a phenomenal question. Bolu, would you like to answer that, or should I go first?
You go first.
So I will just say, bearing in mind that like my own admission was for a PhD, not necessarily like going in straight out of high school. So slightly different admissions process, but the general trends that I can remark on are being able